Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I am Professor Dr. Shahzad Farooq. Today we are going to study the conformational diseases and protein misfolding. At the end of this lecture, student must have basic knowledge of protein misfolding and conformational disease. They should know the biochemical basis and clinical features of Alzheimer disease. And finally, they must have basic knowledge of types, biochemical basis, and clinical features of transmissible spongiform encephalopathies, also called prion disease. Protein misfolding. Protein folding is a complex process that can sometimes result in an improperly folded proteins. These proteins usually tagged and degraded within the cell. However, this quality control system is not perfect and intracellular or extracellular aggregates of misfolded proteins can accumulate. Deposits of these misfolded proteins are associated with number of diseases. Misfolding of protein may occur spontaneously or be caused by a mutation in a particular gene which then produce an altered protein. In addition, some apparently normal proteins can, after abnormal proteolytic cleavage, take on a unique conformational state that leads to the formation of long fibrillar protein assemblies, which are consisting of beta-pleated sheets. These insoluble fibrous protein aggregates and these aggregates called amyloid. Amyloid may be implicated in many degenerating diseases, particularly in neurodegenerative disorders like Alzheimer disease. Alzheimer disease is not a genetically based, although 5% of cases are familiar. The catalytic subunit of gamma secretase are most common cause of familial Alzheimer disease. The dominant component of amyloid block that accumulate in Alzheimer disease is amyloid beta. Amyloid beta is a peptide of 42 amino acids. Amyloid beta, which is 4.3 kilodalton, and as I have already told you, contains 42 amino acids. This extracellular peptide, when aggregated, is neurotoxic, which is a characteristics of the disease, and this neurotoxicity results in the cognitive impairment. The amyloid beta is deposited in the brain in Alzheimer's disease, is derived by the enzymic cleavage from the larger amyloid precursor protein. Amyloid precursor protein is a single transmembrane protein expressed on the cell surface in the brain and other tissue. The amyloid beta aggregates generating the amyloid that is found in the brain parenchyma and around blood vessels. Second biological factor involved in the development of Alzheimer's disease is the accumulation of neurofibrillary tangles inside the neuron. A key component of these tangled fiber is an abnormal form of tau protein, which is hyperphosphorylated insoluble form of normal tau protein. Normal tau protein help in the assembly of microtubular structure, while the defective tau protein block the action of its counter normal part. Apolipoprotein E has been implicated as a potential mediator of this conformational transformation. Exposure of neuron to amyloid beta, which is a neurotoxic, can increase their intracellular concentration of calcium ion. Some protein kinases, including that involved in the phosphorylation of tau, are regulated by levels of calcium ion. Increase of calcium ion may lead to hyperphosphorylation of tau protein. X-ray crystallography and infrared spectroscopy demonstrate the characteristics beta-pleated sheet conformation in non-branching fibrils. Here, formation of amyloid beta 42, which is a toxic and amyloid producing peptide. A key initial step is the digestion by either beta secretase or alpha secretase, producing small non-toxic alpha secretase product and beta secretase product. If the cleavage takes place by beta secretase, we have this product called beta secretase product. And if the cleavage takes by alpha secretase, we have this product and which is called as alpha secretase product. Now, the cleavage by gamma secretase may occur on these product, and the cleavage of beta secretase product by gamma secretase may result in amyloid beta 42, which is the toxic, or amyloid beta 40, which is the non-toxic. If it cleavage occur at this site, we have this product which is non-toxic, but if gamma secretase causes cleavage at this site, we have this amyloid beta 42, which is a toxic one. When gamma secretase act on the alpha secretase product, we have this non-toxic P3. Presenilin 1 and presenilin 2 may be involved in the action of gamma secretase. More precisely, they are the catalytic subunits of gamma secretase. Here you can see the amyloid precursor protein, which is the transmembrane protein. This is the extracellular component. This is the intracellular, and this is the membranous component. This transmembrane protein, called amyloid precursor protein, is encoded by genes on chromosome 21. Amyloid beta peptide aggregate. This is the amyloid beta. Results from the abnormal proteolytic cleavage of this amyloid precursor protein by beta secretases followed by the gamma secretases. And this amyloid beta is a neurotoxic, and when this amyloid beta aggregate, it generates the amyloid. There is spontaneous aggregation to form insoluble fibrils of beta-plated sheets. The scheme of causation of Alzheimer disease, increased production or the decreased clearance of amyloid beta 42 results in increased concentration of amyloid beta 42, and there will be oligomerizations and formation of beta sheets of amyloid beta 42 causing neurotoxicity. Increased level of calcium ion may activate the protein kinase that phosphorylate the microtubule associated tau protein, resulting in the formation of paired helical filaments, results in the neurofibrillary tangles. This lead to deposits of amyloid beta 42 and hyperphosphorylated tau form. Over many years, this process continues for many years and results in the plaque formation and the other neurofibrillary tangles. Both are the important biological features of the disease, and ultimately they interfere with the neuronal functions, resulting in the clinical sign and symptom of Alzheimer disease. If we see the clinical features of the disease include cognitive impairment. Cognitive impairment may be presented as memory loss, or the patient frequently asking the same question or repeating the same story over time and over, patient not recognizing the familiar peoples and places, patients having trouble exercising judgment, or there may be change in mood or behavior, there may be vision problem, or patient is unable to plan and carry out day-to-day tasks. There is gradual loss of memory in these patients. Short-term memory and the long-term memory both are affected, but short-term or the recent memories are more affected. Alzheimer disease is the most common cause of dementia. There may be loss of coordinated movements called apraxia. There may be abnormal gait or difficulty in typing on computer, and the aphasia may occur, which is loss of ability to speech, and then there is lack of awareness in these patients, and they insist that nothing is wrong with him or her. Depression may be there, and sometimes patients present with the aggressive behavior. So there may be a change in mood and personality. If we talk about the treatment, there is no specific treatment. There is only symptomatic treatment. Symptoms such as depression, agitation, anxiety, and insomnia can be treated by appropriate drugs, and antipsychotic may be required if psychosis intervene. Drugs like anticholinesterase have some role, but the most important is the prevention. This can be prevented by omega-3 fatty acids. Now, the transmissible spongiform encephalopathies. Transmissible spongiform encephalopathies or the prion disease. Prion protein is the causative agent of fatal transmissible spongiform encephalopathies. Includes Creutzfeldt-Jakob disease in humans and its variants Creutzfeldt-Jakob disease, which occurs in younger patients and patients present with the early onset of psychiatric or the behavior changes. These also include scrapes which occur in sheep, and finally bovine spongiform encephalopathies which occur in cattle, and this bovine spongiform encephalopathy is also known as mad cow disease. Prion protein cellular, this is a non-infectious form having the same amino acids sequence and the gene sequences as the prion protein scrapes, which is the infectious form. The number of alpha helices present in non-infectious prion proteins are replaced by beta sheets in the infectious form, which is prion protein scrapes, represented PRPSC superscript. This conformational difference lead to the resistance to the proteolytic degradation of these proteins. The infective agent prion protein scrapes is an altered version of normal protein. And this altered version, which is prion protein scrapes, act as a template for converting the prion protein cellular, which is the normal or the non-infectious form, to the prion protein scrapes, which is a infectious form. This prion protein cellular contains predominantly alpha helix, and this prion protein scrapes contains predominantly beta plated sheets, and that's why it is called conformational disease. Prion disease may manifest themselves as infectious or genetic, or there may be sporadic uh, occurrence of this disease. As no viral or bacterial gene encoding the pathological prion proteins could be identified for many years, so the source and mechanism of transmission of prion proteins remain elusive for many years. Human prion protein is a glycoprotein encoded on the short arm of chromosome 20. As I have already told you, normally is a monomeric and rich in alpha helix. Pathological prion protein serves as a template for the conformational transformation of normal prion protein known as prion protein cellular into prion protein scrapes. Prion protein scrapes, which is infectious form, is rich in beta sheets with many hydrophobic amino acyl side chains. When prion protein cellular, which is a normal form or the non-infectious, interact with its pathological form, which is prion protein scrapes, the conformation of prion protein cellular, which is normal or the non-infectious, is transformed from a predominantly alpha helical structure to the beta sheet structure, which is the characteristics of prion protein scrapes. This conformation, which is prion protein scrapes or the infectious form, aggregates into multimeric protein complexes, which is resistant to the proteolytic degradation. Because prion protein scrapes molecule associate strongly with one another through their exposed hydrophobic regions, the accumulating prion protein scrapes units condense to form insoluble protease-resistant aggregates. Prion disease can be transmitted by the protein alone without involvement of DNA or RNA. This is the prion protein scrapes having predominantly alpha helix and very few beta-pleated sheet strands. And this is the prion protein secrepies, which is infectious form. This is non-infectious form prion protein, and this have predominantly beta-pleated sheets. Conformation of these alpha helix transformed into these beta-pleated sheets. Both have same amino acid sequence, but the difference is in the conformation, and this difference lead to the aggregation into the multimeric protein, which is resistant to the proteolytic degradation. Another presentation, here you can see the alpha helix predominantly 40% and two small beta strand segments, but the conversion of this component into beta pleated sheets predominantly, which was undefined here, not mentioned in this picture, and this transform into beta pleated sheets. We can see interaction of infectious prion protein molecule with the normal prion protein causes the normal form to fold into the infectious form. This diagram shows how the exponential increase occurs when the non-infectious prion proteins containing alpha helix interact with the infectious prion proteins containing beta pleated sheets. This prion protein, which is infectious, is called prion protein secrepies, and non-infectious prion protein is called prion protein cellular. You can see this non-infectious interact with this infectious, and we have these two infectious prion protein. These two molecules dissociate and convert into two additional non-infectious prion protein molecule to the infectious form. You can see now these two non-infectious interact with these infectious prion protein and have with this exponential increase. Clinical features includes the rapidly developing dementia, difficulty in walking and change in gait, jerking movement of the muscles. Patient may have hallucinations. Patient may complain of muscle stiffness. There may be confusion and fatigue, and patient may feel difficulty in speaking. Thank you very much. This was all about conformational diseases, and sources of these lectures includes Harper Biochemistry 31st edition and the Lippincott Illustrated Biochemistry 7th edition. Thank you very much.